Good afternoon, good afternoon, good afternoon, everyone. There's a good energy in the room here, but I'm going to have to ask you to settle down so we can get started. So welcome to everybody. Welcome to the front row. My name is Jamie Williamson. I'm a professor in the Department of Integrated Structural and Computational Biology, and I'm your host today. So we got a full house, looks like about 250 in person, and I know we had 7,000 people sign up to tune in virtually. So hi to everybody in the ether. It's great to have everybody here to hear from Alejandra Mendoza. So I wanted to start out by saying a few things about Scripps. So Scripps research started, I was just walking in La Jolla the other day, and south of Prospect is this old building that's now a biotech startup building. And that's where the Scripps clinic started, this metabolic clinic. And Frank Dixon is this person that really brought immunology to Scripps. And eventually they moved up to this campus next to the hospital. And so he came in 1961. And then Gerald Edelman, many people know Gerald as a Nobel laureate, and he presided over this campus, which was, when he was here, the Neurosciences Institute. But he actually was awarded his Nobel Prize for antibodies. So there's this really strong culture and history of immunology from Scripps, and that was continued by Richard Lerner. So Richard was the president for about 25 years. And he presided over a period of unprecedented growth. The size of the institute ballooned, lots of industrial partnerships, hiring faculty. He had the vision to bring chemistry to Scripps, which has been a huge asset ever since that happened. He started the graduate program, and he had an active lab the whole time he was here. Groundbreaking immunology, and just perhaps to highlight something, is he developed Humira, which is a treatment for arthritis. So immunology continues. In the 2000s, we had a couple of really major accomplishments by our scientists here. So Hugh Rosen and Ed Roberts developed what is now called Zeposia for multiple sclerosis. And then many of you have heard from Andrew Ward, who gave a lecture here at the front row. And Andrew really was visionary. He patented the spike protein antibodies, and it turned out to be essential for developing the COVID vaccines. So the strong tradition continues. And so there's a lot of activity that's ongoing. So Pete Schultz is the president. We have the Caliber Institute. They're focused on infectious disease. They've got new drugs that they're developing in many areas. And then we have, like Dennis Burton is working on neutralizing antibodies for HIV to try to make an HIV vaccine. So immunology is very much, it's at the core of the Institute since it started, and it continues to this day. So how do we do this? Well, we're a not-for-profit institute. We have about 150 faculty members. There's probably another 1,500 scientists that are students and postdocs and staff scientists that fill the labs and help carry out this work. We also have an amazing graduate program. So the way that we do this is primarily funded by grants from the government, but we also rely on major gifts in philanthropy and partnerships with industry and individual donors. And I know many of you here might be in the legacy circle or the scientific something. Sorry. Thank you. And Science Trust. Thank you. Sorry. I need to work on my intro a little bit. So we do this, and you really need to understand there's no gift that's too small. I mean, it really takes a coalition of people to support the Institute. And, all I can tell you is that the scientists here are just working really hard and are very deserving of whatever support that you can offer. So here we are with Alejandra. And so I served as 7 years as the academic affairs vice president, and by far the best thing that I got to do was hire faculty. So we hired Alejandra in 2022, and actually she was the last faculty member that I onboarded before I returned to my lab to do full-time research. So it was just remarkable to sort of interview all these people, and then you settle on someone like Alejandra, who's obviously going to be a star, and she also fits right in to the program at Scripps. She's working on immunology, but the area she's working in is the crosstalk between the nervous system and the immune system. And we call them nervous system, immune system. They're systems. Are they separate? But what's remarkable about human biology is we're still learning many things and about the crosstalk between those things. And that was the subject of the program. She got a major NIH grant funded recently to study this, and it's going to be the subject of our lecture today. And now, at the end, I'm going to come back, and we're going to have a little Q&A. So at any time, if you're in the virtual audience, you can type into either the chat or the Q&A a question, and they'll be relayed to me at the end. For those of you here in the physical audience, stick your hand up, and we'll have runners. It's a little hard for me to see, but we'll have runners with a lightsaber, and then they'll draw my attention to call on you, and then we can have questions from the audience. So I encourage you to do that. It's a lot of fun. And I'll come back at the end and we'll discuss the lecture. So without further ado, I want to introduce Alejandra Mendoza.
All right. All right. Thank you, Jamie, for that very nice introduction, and thank you, everyone, for making time out of your day to come and see what's happening at Scripps. So as Jamie mentioned, I'm going to talk today about the intersection between the immune and nervous system and how we sense danger and how these crosstalk between the nervous system and the immune system coordinates inflammation. So before I get started, I wanted to give you a little bit of my personal background on how I ended up here at Scripps. So I was actually born in Stillwater, Oklahoma. I know that sounds kind of odd, but I was born in the Oklahoma State Hospital, Oklahoma State University Hospital, because my parents were both going to school there. And after I was born, a few months after, we actually went back to Venezuela. So both my parents are Venezuelan, and we went back. And I grew up there in the capital in Caracas. And my parents are not scientists. No one in my family is really a scientist, but I developed this love for science very early on. The classes that I gravitated towards in school were always science, but honestly, I didn't know that being a scientist was a real possibility. But then by the end of high school, doing some digging, I thought, well, maybe this is something doable, and it is something doable, maybe not in Venezuela, but coming back to the United States seemed like a good idea to do this. And so I really got my way in to a career in science through this meeting with Michael Gaines at the University of Miami, who ran the undergrad research and community outreach office, and he ran a program for students from underrepresented backgrounds to be put in contact with science mentors. And this really opened up this opportunities for doing science. And so that's how I ended up going to the lab of Gary Kleiner at the Miller School of Medicine at the University of Miami. And I worked there as an undergrad for 3 years. And I also did a summer program at New York University and the Scripps Institute one summer with Juan Lafay, and this was really the biggest introduction to immunology. I actually got to learn what regulatory T cells were in this lab, which I'll come back to. I guess I've circled back to them. And I loved New York. I also loved the Scribble Institute, so I decided to go to grad school there, and I joined the lab of at the time, a new assistant professor, Susan Schwab, and for my postdoc, I stayed in New York and went to the lab of Alexander Rudensky. And in both of these labs, I studied how cells from different lineages interact to mediate immunity. So in Susan's lab, I worked on how lymphatic endothelials and T cells interact for T cells to move around the body and also to mediate their survival. And in Sasha's lab, I looked at how effective networks are established between T and B cells to mediate proper B cell immunity, and then this neuroimmune talk, which is going to be the topic that my lab is heavily invested on now, and the story that I'll talk about today. I'm glad that Jamie mentioned that he onboarded me, because when I was looking for a place to start my lab, when I got the offer from Scripps Research, I was very excited. I mean, this place is amazing. If you look at the faculty, there's so many amazing prizes, because of course the faculty are great here. So we have 6 Wolf Prizes, 6 Nobel Prizes, 4 Howard Hughes medical investigators, and as Jamie mentioned, many FDA-approved drugs have come from discoveries that have been made at Scripps. And the graduate program is one of the top 10 in the country, which for a starting assistant professor was a huge, important deal. So the question wasn't so much when I got the offer from Scripps whether I would take it as where do I sign? And I literally have a picture of me signing my offer letter after getting off a Zoom call with Jamie. And so, yeah, in November of 2022, I made the trip from New York to San Diego and started our lab right here. After renovations and stuff, the lab started really, it opened up in March in 2023, and I started with a very small group of amazing people, and now we've grown to be a much bigger group of amazing people too. So, okay, so that's it for the personal stuff, and we can talk about science. So for everyone to get on the same page, I wanted you to think about how many, from the time you woke up, or even from the time you arrived in your car and to the time you sat down on your seat, or if you're at home sitting wherever you are, how many stimuli have you received from the time you, again, woke up, or in the last few hours? It's probably too many to count. You've touched many things. You've smelled, well, a bunch of stuff. You've been exposed to the humidity, the light, the UV radiation. So all of these different things, we are constantly bombarded by stimuli. And some of these are things that we are able to consciously detect, and a lot of them are not. Things like microbes are also you're exposed to constantly. And our bodies need to be able to sense all of these things, again, conscious or not, and mount responses and adapt to them, because our bodies depend on maintaining internal stability for our organs to function. And this is because cells and which make organs function properly at optimal ranges of things like temperature, sugar, molarity. So, and these parameters we usually refer to as set points. And deviations from these can impair how enzymes work, and meaning how your cells function, how your cells communicate. And because of that, these deviations can be harmful and induce tissue damage. So I'll say this word homeostasis over and over today, and I hope you remember what this is, because it's a very important process. It's the active process that we do to maintain a set point for all of the parameters that we care about. Again, this is an active process, and it is essential for us to maintain these internal conditions despite all of these environmental perturbations that we're constantly exposed to. And this is what really makes us resilient to all these different environmental fluctuations that we're constantly experiencing. And so the way this works is a very basic homeostatic circuit. And the easiest way, or I think the most intuitive way to think about this is how a town deals with a fire. And I'm sad to say that in California we think about fires a lot, so it's not, I didn't do it because of that. It's just because it is like a very easy concept to grasp from this. But in this homeostatic circuit, the inducer would be the fire. And so there's factors associated with the fire that we can detect. So things like smoke and heat, that can be picked up by a sensor. In this scenario is a smoke detector. And that then turns on a mediator, which is a fire alarm in this case, a phone call to the fire department, that then induces effector mechanisms that can deal with the perturbation that's the inducer, which is a fire in this case. And effector mechanisms are usually, there's many different things that get induced in order to deal with this perturbation. So things like not only calling the fire department, but making sure that the water is routed in the correct way so that it gets to where we want to have it, closing roads so that we're able to have the fire department get there and not have harm in other people. So all of these things work together in this sort of circuit to ensure that we are at homeostasis. And when we think of inflammation, how many people, so if we do a quick show of hands, how many people think that inflammation is a bad thing? Okay, it's a mix. A lot of you have done your homework and think that inflammation is good. Good. So it's a mix, right? So I think it gets a bad reputation, or a lot of it comes from its name. Inflammation literally means to set on fire. And in actuality, so it would be tempting to put it, when we think about it in this context on the circuit, to put it as inflammation would be this, like this inducer of disruption or this perturbation. But actually, inflammation is the process that tries to get us back into a homeostatic once we have either an infection or a damage of the tissue. And so rather than being, again, the fire, is the whole process. And so the way this works is that a pathogen or injury will be the inducer. Immune cells that are equipped to sense patterns associated with damage or with pathogens themselves, then sense that there's a pathogen, and then trigger mediators' responses to start an effector response. And mediators in this case are molecules that immune cells make or activation states. And then effectors are the cells who actually are induced to, let's say, for example, to kill this pathogen that has come in. And so I said, well, inflammation probably gets a bad reputation from its name, but it is a little deserved because it does come at a price. While we need it for getting rid of pathogens, and it is important that we induce these responses, one, it takes energy. So we usually, when we're fighting infections, we do intentional trade-offs. So we will suppress things like growth and reproduction just so we have the resources to mount an immune response, which again takes energy. But it also induces a lot of collateral damage because killing pathogens, killing infected cells induces tissue damage. And so too little or too much inflammation is a problem. Too little will leave us defenseless against pathogens and damage, and too much can induce a lot of damage and these trade-offs that may suppress processes that we would still want, like growth. So my lab studies basically this question: how do we manage this balance? How different cells in the tissues that interface with the outside, which are exposed to all these different stimuli, some which are okay, some which are beneficial for you, like nutrients, and some that are pathogens and bad for you. How do we, at this environmental interfaces, how do cells work together to maintain homeostasis? And so in very general terms, what the lab tries to do is try to understand how the location of these insults, the intensity of them, and as well as the type of pathogens and or inflammatory agents that we see induce cellular states and gene expression changes, and how cells communicate with each other to make sure that it all works to maintain homeostasis at these barrier sites. And our lab works in several of these sites, so we have students and postdocs working in the lung, the mammary gland, the intestine, and the skin, which will be the topic for today. And they will be outside on a table, so everyone, I didn't force them, I promise, but they're all here, so I think they're more than happy to tell you about their work. So make sure, if you can, go talk to them after the talk. Okay, so, all right, so today I will be focusing on the skin. So the skin is, I think, one of the most interesting organs because it is not only is it our physical barrier to the outside world, it is, and it is a living barrier. And again, it's constantly exposed to all this different stimuli, some which is noxious and some of it which is benign. And because of this, of it being at the interface with the environment, is the site where most pathogens come through. And because of that, it is associated with a very large number of diseases. And again, this balance of too much inflammation and too little inflammation is very important to be maintained in the skin. Because again, too little inflammation leaves us prone to infection. And here I just listed a few infectious diseases and cancers, which are, again, maybe failure from the immune system to clear. And then diseases that are more associated with too much immunity, like inflammatory or autoimmune diseases like psoriasis, atopic dermatitis. And so this is a very narrow list. There's way more skin diseases than I listed here. But again, the main point that I wanted to drive across is that because we need to maintain this very careful balance, it is easy to tip it one way or the other. And so trying to understand what mechanisms maintain this defense and tolerance balance is really important. And then I just wanted to very quickly point out that this matters not only as interesting biology, but it matters for a lot of people. Over almost 5 billion people and worldwide suffer from skin diseases ranging, again, from infectious diseases to chronic inflammatory diseases. And this is not counting the over 6 million people who have cancers, so skin-associated cancer. So it is a major burden on us and a worldwide problem. And if you see California here, I know it's kind of hard to see, but there is a lot of high incidence of all of the skin diseases as well as skin cancer. Okay, so what the topic for today is how do we regulate, how do the nervous system and the immune system interact to regulate skin immune homeostasis. So again, I won't say this point again, but we have all these stimuli. And we have, the skin is rich with immune cells that patrol the tissue that sends patterns associated with damage and inflammation, with damage and pathogens, and can activate immunity to protect us when there's been a breach in the barrier. And we also have, in addition to those immune cells, we also have this rich network of sensory neurons that innervate the tissue. And they induce protective responses, things like avoidance when something is noxious to you, and things like even like itch, which it does sound like it is uncomfortable, but there's a physiological reason for it, right? We want to dislodge things that are bad for us, that are noxious on our skin, or maybe like a mosquito or something like that, where we want to avoid that threat. And so the two systems protect us, and it is maybe no surprise that they interact and that these interactions go beyond just inducing protective behaviors. So for a long time, we've known that molecules made by immune cells can induce responses in sensory neurons to heighten pain and itch. But what has come to light in the past 10, 15 years, and we understand much more of now, is that neurons themselves can talk to immune cells, and this is important for inducing inflammatory responses. And what does this mean? So it means that neurons can make signaling molecules, for example, like a neuropeptide, that can be sensed by immune cells, and that triggers activation and recruitment of immune cells, and it's important for inducing a proper immune response. So given that I just told you that immune cells can induce pain and itch in neurons, and I told you that neurons can induce activity of immune cells, then that ends up in a very bad combo. Because if we have immune cells that can activate sensory neurons, and those sensory neurons can activate immune cells, then you can see how this forward loops of pain and inflammation can turn into chronic maladaptive states. And so if these things are true, then there must be negative regulators preventing this feed-forward loops driving chronic inflammation. And so when I was a postdoctoral fellow in the lab of Alexander Rudensky, I was really interested in whether regulatory T cells could play a role in this, in providing this negative feedback on the neurons themselves. And the reason why I thought maybe regulatory T cells would be important in this process is because these cells do many different things, but they are incredibly important for modulating immunity. There's less regulatory T cells in your body than almost any other cell, yet they have a very important job, because without them, so mutations in the gene, in a gene called FOXP3, which is required for their development and function, mutations in FOXP3 are induce this fatal systemic autoimmunity. And if you look at this graph here, with patients that have mutations in FOXP3, most patients have died by the time they're 20. So these cells are quite important. And I would be remiss if I don't talk a little bit about them, about their history, because this year there were some big news about regulatory T cells. So for a long time, so they were described in the late 60s. So in the 70s and 80s, people talked about suppressor cells, and this was kind of like the dark days of regulatory T cells. We don't like to talk about those because we didn't have good markers for them. We didn't really know how they worked. We didn't know who they were. And so it was really until the 90s where markers for them were found, and we could do transfer experiments and things like that, and saw that they were very important for immune suppression. And then in the 2000s, a lot of, with the discovery that FOXP3, the gene that I mentioned earlier, was important for their development and function, came a lot of functional studies linking this autoimmune disease that I mentioned, IPEX, with lack of regulatory T cells. And then we had, we could use mouse models to test the function of these cells. And then fast forward in the last 25 years, so from the 2000s till today, there's over 40,000 publications on regulatory T cells. There's over 200 trials trying to use these cells for disease management. And this year, the Nobel Prize was awarded to the discovery of them, to the people who I've highlighted in red. And for those of you who did not tune in from Howard Hang's front row lecture last year, actually Shimon Sakaguchi, who's one of them, was at Scripps for a couple of years as an assistant professor before going back to Japan. So going back to our story, so we thought, or I was really interested in whether these cells had any interactions with neurons to dampen this forward loops of inflammation. And this work was started, so I started it as a postdoc when I was in the Rudensky lab, and I had help from Paolo, Regina, Emma, and Stas, and Sasha, and the Rudensky lab. And then 2 of my postdocs now here at Scripps also worked in the story to get it finished up, and it was recently published a few months ago in September. Yeah, a few months ago. So yeah, so I'll give you a little glimpse of that story, because this was really what kickstarted how this interest in neuroimmune interactions, at least for me. Okay, so we thought, so the reason we were really interested in whether they had this crosstalk with neurons is that in the skin, we very often found them near axons innervating the skin. And so if we just plotted the distance of a Treg to the nearest axon, we saw that very often these cells were in direct contact, so about 30% of them were, again, directly touching. And then most of them, the ones that were not touching, were within 20 microns, which is, I put into context here, kind of the size of a cell, so that's about like 1 to 2 cell distance. So they were close enough to maybe mediate responses or some sort of crosstalk by either direct contact or maybe by a secreted factor. And so to test whether this could be the case, we used a genetic model that the Rudensky lab had developed in 2007, where we could deplete regulatory T cells specifically, because using the human diphtheria toxin receptor expressed in the FOXP3 locus. So again, only regulatory T cells would express this receptor, which when it binds diphtheria toxin, it inhibits protein synthesis in the cell, and it causes cell death. So this is a nice way in which we can ablate these cells very fast. And then again, the Rudensky lab showed in this paper in 2007 that if you get rid of those cells for a long time, the mice actually succumb to death because they develop this fatal autoimmunity. But I wanted to try to deplete them very, very quickly before there was any onset of autoimmunity, and then see if there were any effects on the nerve, on sensory perception. And so we waited 18 hours, which is a little bit overnight, and we saw for all of the immune parameters that we saw, we couldn't see any increased inflammation yet. So this seemed like a good time point to look at. So again, the only thing that we could see really being changed here was the cells being gone themselves. And so to test whether there was any changes in sensory perception, we used these established assays to look at how the mice were sensing different types of sensory stimuli. And this is usually referred to as nociceptive behavior. So we looked at how the mice sense heat sensitive, or how sensitive they were to heat, by putting them on a hot plate that is at 56 degrees. So that's uncomfortable. If you touch something at 56 degrees, it feels uncomfortable. And the mice do this very stereotypical behavior where they like jump and then they lick their paws to try to mediate that or to try to dampen that heat that they sense. And so what we measure here is how fast it takes for the mice to do those defensive behaviors. And what we see is that when we get rid of regulatory T cells for a short amount of time, they respond much faster, suggesting that they sense heat much more readily. We also did a version of these assay for cold sensitivity. So in this case, we put acetone in the paws of the mice, and acetone feels cold. So for anyone who's had acetone in their skin, anyone that uses a nail polish remover, for example, will know that acetone feels cold. And if you want to sense this, actually the lab will have acetone outside to put on your skin. They didn't let me do a hot plate. They said there was some liabilities, but anyway. Okay, so in this assay, we measure something different. We measure how long the mice lick at their paws to try to get that acetone off, to like remove it. And what we see is that when we get rid of regulatory T cells compared to controls, the mice do that licking behavior more, suggesting that they're sensing the cold for longer. And then we also tested how the mice sense mechanical stimuli by giving them different strength of filaments, and then seeing how often they retract their paws when they see the different strength of filament. And what we saw is that the line here in black is the mice that were depleted of Treg cells, and they respond much more often and to thinner filaments than the control mice, suggesting that they sense mechanical stimuli much more strongly. And so how is this happening? Why are they're not modulating inflammation to modulate the sensitivity? So what are they doing to modulate sensory neuron responses? And so we looked at gene expression data to try to find some candidates, and we thought that there was one very enticing candidate, which is called, the gene is called PENK. It codes for proenkephalin, and proenkephalin is the proform or the precursor of enkephalins, which are these small neuropeptides that bind the opioid receptors. So these are endogenous opioids that your body makes to dampen pain perception. And so this, and these receptors, the opioid receptors are the ones that bind drugs like morphine or fentanyl. So given that a population of regulatory T cells express this neuropeptide, it seemed like this was a good candidate. So, and in fact, the regulatory T cells that expressed this neuropeptide were highly enriched in the skin. So again, it seemed like a good candidate. And so to test whether this neuropeptide, this endogenous opioid from regulatory T cells was doing anything to sensory perception, we used a genetic mouse model where we delete the gene PENK only in regulatory T cells. So every other cell that makes PENK can make it. Only in this case, regulatory T cells cannot, and those are the mice here in blue. Compared to their litter-mate controls. And we did the same battery of assays to test sensory perception in these mice. And what we found was that those mice were also more sensitive to heat. They were also more sensitive to the mechanical, they had higher mechanical sensitivity. But very interestingly, they did not care about the acetone at all. So this mechanism of cold sensation that regulatory T cells mediate is independent from this opioid production from regulatory T cells. And now the question was, okay, so if this neuropeptide production by regulatory T cells is preventing this forward inflammatory loop, then you would expect that it would reduce inflammation. And so what we did for these experiments was to induce psoriasis in these mice. And again, these are mice where we've deleted PENK only in regulatory T cells, and we induce psoriasis in the ear, and what we see is that the mice do develop worse psoriasis, so we see that the mice have more immune cells infiltrating the ear. Here we're showing neutrophils. They also, the tissue swells more, so compared to the controls, and if you look at the histology here, I hope that you can see that there is way more inflammation in the mice where we've deleted PENK in regulatory T cells compared to controls. So from these experiments, it seemed like regulatory T cells were dampening these responses in sensory neurons as a part of, or as a new mechanism to dampen inflammation. So how are they doing this? So sensory neurons are very diverse. There's at least 12 different types, and so to kind of get a sense of what was happening, we decided to turn to gene expression, and we did single-cell gene expression in sensory neurons that innervate the skin to be able to look at all of them at once. And what we found was that several populations of sensory neurons had gene expression changes, but one particular population, which we found the most interesting, had this enrichment of genes that are associated with inflammation. And so we think that this is probably, and that those also express the receptor for, one of the opioid receptors for enkephalin. So it seems like this is probably the cells that are responding to this. Of course, as I've mentioned before, inflammation causes pain. We've known this for centuries. This has been described since the first century, but we always thought, and if you read any review on regulatory T cells and pain, we always thought that they would regulate pain by regulating inflammation. And so what we found was quite surprising, that it's actually the other way around, that Treg cells seem to modulate neuronal activity by secreting this endogenous neuropeptide to act as a cellular analgesic to decrease pain, and that by modulating pain, these cells actually suppress inflammation. And this is interesting biology, but it also means there's room here for therapeutic intervention, because we can potentially use this phenomenon and immune cells and this crosstalk with the sensory nervous system to try to prevent exacerbated inflammation and pain responses. And so, again, this is something that the lab is interested in doing. We would like to target this Neuro-Immune Crosstalk to break this pain and inflammation forward loops. And one of the great things about being here at Scripps is that we have lots of amazing chemists with whom we can do experiments, and we are currently planning on doing a high throughput screen with Luke Lerson's lab here at the chemistry department to find molecules that target this axis for modulating pain and inflammation. And the lab is also just interested in finding more because, again, there's many different types of neurons, there's many different types of inflammation, which means that there's lots of room for preventing this neuroimmune crosstalk that can mediate disease and chronic inflammation and pain. And so to do this, I'll tell you, and I promise it's going to be 2 minutes, what the lab is doing now. So we are looking at, again, gene expression at a global level in sensory neurons and how they respond to inflammation and to different types of inflammation. And this is because not only are sensory neurons diverse, inflammation is very diverse too, because different types of pathogens induce very different types of inflammatory responses. And so what we're trying to do is understand how different sensory neuron populations respond to different types of inflammatory stimuli. And I'll give you a little glimpse of some of the things that we've found, which is, so if we look, so each color here is a different type of inflammation stimuli, and every row is a different population of sensory neurons. And what I hope you can appreciate from this pattern where you see different circles, so the circles indicate changes in gene expression. So what we see is that different neuronal populations have different changes in gene expression in response to different types of immune perturbation, and these are also time dependent, so which again means that there's lots of room for us to investigate and also therapeutically target. And one way that we could do this is to also try to harness this. So I've talked about how these pro-inflammatory loops are so bad, but actually we can use them when we want to fight something like cancer. So do we see something similar when we look at, for example, tumor responses? So we have found, and many others have also shown this, that tumors themselves can get innervated, and this usually is a bad prognosis. But what we have found is that if we look at gene expression changes in neurons, on sensory neurons, we find that early in the tumor and late in the tumor, we see different changes in gene expression in different populations of sensory neurons. And Susanna, the postdoc who works in this, who again will be at the table and she can tell you a little more, she has found that actually by manipulating the activity of the neurons that are innervating the tumor without doing anything, without giving them any chemotherapy or any checkpoint blockade or anything, she can induce the tumors to shrink. And this is very preliminary, but very exciting, so I wanted to share with people. Okay, so with that was my last data slide, so wanted to refresh you on the key takeaways that I hope you remember once you leave. The most important thing is that homeostasis is a very important process for all of your tissues to function, and that your barrier tissues which interface with the environment have to always balance this protection and tolerance, because inflammation is both good and bad. It's protective and destructive. And that your sensory nervous system is an active participant in inflammation, not just a bystander. And that this communication between the nervous system and the immune system can hopefully be targeted for opening up new therapeutic paths to treat chronic diseases associated with pain and inflammation. And so I will finish by thanking the people I mentioned, the people when I was talking about the work, but a brunt of the work was done in the Rudensky lab when I was a postdoc, and I mentioned Stas, Paolo, Regina, and Emma who helped me there. We also had some collaborators at the University of Bonn and Weill Cornell that provided us with some mice and also helped us do some of the experiments. The people in my lab, so Susana and Sebastian are the ones that were involved in the work that I described. But again, go talk to the other people in the lab who also have very cool projects. The sources of funding from my lab, and of course all of the people at Scripps who make this place run. It really does take a village, and so we are very grateful that they do such a good job running this place. The graduate school, again, is fantastic, and our students are amazing. And lastly, I wanted to say a special thanks to Stas Deki, who is my partner in science and in crime. So thank you, because we bounce ideas off each other a lot, and he also helps a ton of my students. So thank you, Stas. And I also wanted to mention very quickly that this place is one of the big, most important and amazing things about Scripps is the amount of collaborations we have. And so I listed just the people that we have collaborations ongoing at the moment, and I was shocked to see that I have collaborations with every department at Scripps, which was like nice to see. And then I also wanted to give a special thanks to the Department of Immunology and the Department of Neuroscience, who have been amazing to be involved with. I've infiltrated, I'm like the dirty immunologist who've infiltrated the neuroscience department, and they're so nice about it. And lastly, I'm so glad that Scripps does this front row lecture is so nice to engage with the community, and so I'm really happy that the executive office sees value on this, and of course, and thank you, Jamie, also for hiring me, but also for hosting this. And then, of course, everyone who tuned in at home, everyone who made it here, thank you for caring about science, and your curiosity and your support is what makes discoveries possible. So thank you.
Thanks so much, Alejandra. Please, let's have a seat. You know, one of the things that's really gratifying is when you made a good decision. It was a good decision to hire you.
Nice. Hopefully you think so for a while.
So I'm going to ask, I got a question relayed to me from the virtual audience. I had the same question. Your logo, and it's evocative of Henri Matisse cutouts. But could you explain? I have a theory, but I think we should just hear your version of it.
Yeah, so it is a triangle, and there's 3 different cell types in there. There's a stromal cell, so it's like an epithelial cell, a neuron, and then a T cell, or an immune cell. And the idea is that there's 3 things that we care about and how these cells interact. Is the context, the type of insult, and then the place. So this, like, how this all works together to mediate homeostasis.
Nice.
So.
Good logo.
Thank you. I made it.
So I'm going to ask a really naive question, but I'm pretty sure people are kind of wondering this. But so you talked about the relation between sensation of pain and inflammation. And we all take ibuprofen, we all take naproxen, some of us take opioids, hopefully in moderation. But, so what, do taking these medications really, do they actually interfere with your, immune sensing? Do they make you more or less susceptible to infection and, autoimmune conditions?
Yeah. So I think about this a lot. So yes, I mean, the short answer is yes. And I think with a lot of the new biologicals, so again, for skin disease, this is a major problem. So for chronic inflammatory diseases, we take this immunosuppressive drugs, and people do have this increased incidence of infections later. And with any immunosuppressive drug, we do this. So opioids are interesting because usually we don't take opioids to reduce inflammation, but we do take them in response to pain induced by inflammation. And actually from mouse experiments that we did, we do see that opioids themselves can dampen inflammation alone. So I would not recommend taking those, obviously, for inflammation, but it does mean that we can harness the signaling to maybe make drugs that target both.
Right.
And hopefully something that's not
that's a great
an exogenous opioid.
Yep, that's great. So do we have any questions here from the, I don't see the lightsaber. Yes, right down here.
Just a follow-up. So you're saying that taking like ibuprofen to like for arthritis pain to reduce the swelling, which damages the joints, actually is not a good thing because it makes you susceptible then to infections later on in life?
No, sorry for, I did not mean it. I did not mean to say that was, so the ibuprofen, aspirin, those target very late stages of inflammation, and we haven't done any experiments really to see like how I mean, they dampen inflammation for sure, but usually this is, again, for things like arthritis, this is damaging inflammation. So here we're putting the brake. For other conditions where we're targeting things like increased immunity, things like psoriasis and atopic dermatitis. So for these diseases, we use biologics that are also dampening of immune responses. And this is where I'm saying that there's like this increased incidence of infections so, and again, more often than not, being immunosuppressed is better than being autoimmune. You can live, and regulatory T cells are actually a good example of that. You can live without all your T cells. You cannot live without your regulatory T cells because having this like destructive inflammation is worse for you. So I would say if I had to take the trade-off between immunity and autoinflammation, I would always like take a hit on immunity.
Sure.
Comment on prednisone and this whole interaction of...
Yeah, so I mean, I'm not really a medical professional, so I would rather abstain from that, if that's okay with you.
Yes, neither of us are an MD. So yeah. I have a question from I'm going to aggregate a couple points from the virtual audience. But so you talked about pain and itch. But are those really the same pathway, and do they interact in a different way with the immune system and inflammation? Yeah,
so they are not. They are mediated, as far as we understand it, they're mediated by different neurons, and the mediators that drive the pain sensation and itch sensation are likely different, though there might be some overlap. But they are different neurons, and it is so one of the things that I think about a lot is how for a lot of the skin conditions, things like psoriasis, eczema, contact hypersensitivity, all of these diseases have very distinct types of inflammation, yet they all induce itch, and a lot of them are triggered by very similar stimuli, which is usually things like sunlight and changes in temperature and anxiety. So there are some commonalities in the pathways that will drive these flare-ups, and likely maybe the sensory neurons have an effect on this, but they are separate.
So at the beginning of your talk, you've outlined your lab, and you're working on all these other tissues. And so are the T-regs that operate in these other tissues the same? Are they different? Or is there crosstalk between them? Are they moving around?
So all of the most of the immune cells that are present in tissues outside of the lymphoid organs, so like things like the lung, the gut, they adapt to tissue, so they look very different. Mm-hmm. And so, for example, the regulatory T cells that have enkephalin were very enriched in the skin, and there were some also in the gut, but they are slightly different because they do have this different adaptation to tissue, and the frequency of them is quite different. and this is true for other immune cells as well. Like, for example, macrophages are very different depending on where they are. The biggest example is, for example, microglia, which are in the brain. Those macrophages look completely different from your Langerhans cells, for example, which are in your skin.
Okay, so we have a question up here.
You sort of got my interest when you mentioned cancer, and this might have some possibilities. Could you repeat what you said, because I didn't quite understand the mechanism?
So we're still very much finding this or trying to understand that ourselves. What we are seeing is that neurons innervate tumors, and we see that mice with tumors have changes in gene expression in sensory neurons as the tumor progresses. So to some extent, our bodies are sensing that there's a tumor at a sensory capacity. Whether we can use those neurons that are infiltrating the tumor to promote immunity to the tumor is what we're trying to understand. And also, I mean, they might have a pro-tumor role too. They likely have. And so what we're trying to understand is whether different types of sensory neurons do different things, so then maybe we can target neurons that are, maybe activate neurons that are, that dampen tumor growth, but increase anti-tumor responses, for example, or the opposite for tumor, for innervating populations that promote tumor growth.
We have, let's see, it's a little hard to see, but I don't see. So I actually noticed this too. In one of your slides, you had, where you were doing the mouse behavior, you had a little sex thing.
Yeah.
But you didn't mention the sex. And so it's important to always include sex as a variable in experiments. So was there a sex difference?
There is, for sure. So, so in basally males and females, when you do behavioral experiments, they have baseline differences. And specifically for regulatory T cell ablation experiments and this opioid connection, there was an effect. It was funny. So for heat, the males were very sensitive. The females did not care at all. For mechanical sensitivity, the females cared way more and the males less, and same for the cold. So it's really, it is why we have to do experiments on both and control always for sex and like compare both to the chagrin of our students. But
yeah,
I
mean, but is there anything obvious in the T-regs that's
you mean
different?
if there's...
Is there a sex difference in that level, or, you know, or is it something higher order?
We could, so we tried very hard to find anything other than the behavior itself that was different in males and females, and we couldn't find it. So immunity wasn't different at all. I mean, that didn't change, but whatever way we looked at it, we couldn't. We didn't see more. When we looked at activation in the neurons that innervate the tissue in response to inflammation, we couldn't see any increase or change in the activation of them. So it seemed to be something at the behavior level rather than the activation of the neurons themselves. But we don't know, and this is a very interesting question.
Right, okay. We're kind of up on the hour. So this is actually our last front row of the season. So this is the end of season 8, if you can believe it. And so we're going to take a break. We will be coming back. We don't know what the program is yet, but our first front row next year will be in February. So thanks to Alejandra. I mean, this was I learned a lot. I hope you did too. It was very clear, very exciting stuff. So stay tuned. You're all on our mailing list. You'll find out when the next front row is in February. And when you come, you'll find me in the front row.